Good day. Myself S. Periyar Selvam, working as an associate professor in the Department of Food Process Engineering, SRM Institute of Science and Technology, Katangulattur. Welcome to the Department of Food Process Engineering YouTube channel. In the current session, I will brief about the Sauerkraut fermentation. Sauerkraut fermentation is a natural fermentation where shredded cabbage mixed with salt undergoes a lactic acid fermentation, transforming into a tangy preserved food. Sauerkraut means sour cabbage in German. Sauerkraut is also called as acidic cabbage. Natural fermentation occurs through endogenous bacteria present in the cabbage in the presence of 2 to 3% of salt. The major product of sauerkraut is lactic acid, and sauerkraut provides specific flavor and texture due to the presence of major and minor component present on the sauerkraut. A vegetable fermentation will undergo a spontaneous lactic acid fermentation under the appropriate condition or specific condition. The natural microflora present in the plants are anaerobic microorganism, aerobic microorganism, coliforms, lactic acid bacteria, yeasts, and molds. The anaerobic microbial load is 10 to the power of 5 to 10 to the power of 6. Aerobic load is 10 to the power of 6 to 10 to the power of 7. Coliform load is 10 to the power of 4 to 10 to the power of 5. Lactic acid microbial load is 10 to the power of 1 to 10 to the power of 3. Yeast load is 10 to the power of 1 to 10 to the power of 3. Molds load is 10 to the power of 1 to 10 to the power of 3. As I mentioned before, sauerkraut is a natural fermentation. No heat process is required to control or inactivate the undesirable or unwanted microbial growth. The fermentation is carried out by minor populated lactic acid bacteria, whereas dominated and successfully carried out the fermentation process. The mixed microbes are involved in the fermentation process. It is initiated by a specific group of microbes for a particular period, later carried out by other microorganisms due to the accumulation of inhibitory components. The steps involved in the sauerkraut fermentation are harvesting, washing, trimming or removing unwanted part, shredding or sizing, later added into the brine solution or salt solution for controlling the undesirable microorganism or spoilage microorganisms, later incubated for 20 to 30 days. During the period, cabbage dehydrated and production of lactic acid and other minor components. After the fermentation, processed and packed for further uses. The key point should be followed during the sauerkraut fermentations are the cabbage and salt mixture is weighted down to squeeze out the juices and incubated at room temperature in covered container. The container incubate the entry of oxygen into the mixture and allow or maintaining the anaerobic fermentation condition. At the end of the fermentation period, the pH should be approximately 2 and the sauerkraut should containing at least 1% of lactic acid. Throughout the fermentation, it is critical to prevent the entry of oxygen. If the oxygen entry or the presence of oxygen could permit the growth of spoilage microorganism, particularly acid-loving microorganisms such as molds and yeast will grow and spoil the sauerkraut fermentation. Sauerkraut fermentation occurs through the mixed microbial population. It's initiated by Leuconostoc mesenteroids and continued by Lactobacillus plantarum and Lactobacillus brevis. Leuconostoc mesenteroid initiated the fermentation process and dominate the microbial population, and it grow well at 21 degrees Celsius and produce the mannitol. The Leuconostoc mesenteroids not inhibited by 2.5% of salt. In the later session, it's continued by the Lactobacillus plantarum and produce the lactic acid reaches up to 1.5 to 2% with no gas productions. And also Lactobacillus plantarum remove the mannitol. The mannitol is reason for the bitter flavor. The residual sugar and mannitol fermented by Lactobacillus brevis and increase the lactic acid concentration up to 2.4% and provides the bitter acid flavor. Fermentation will be stopped and canning and refrigeration will be continued after the fermentation. High quality of sauerkraut containing high amount of lactic acid, that is 1.7%, and a low level of flavoring component, that is a diacetyl. As I mentioned in the previous slide, sauerkraut fermentation occurs through the mixed microbial population. It's the group of lactic acid bacteria or species involved on the fermentation process. It's initiated by the heterofermentative lichenostrok mesenteroids, followed by the heterofermentative rods such as Lactobacillus brevis and homofermentative Lactobacillus plantarum and Pediococcus cerevisiae. Livestock mesenteroids has relatively short lag phase and rapid growth occur over a wide range of temperature 15 to 21 degrees Celsius. Livestock mesenteroid gas forming and heterofermentative microorganisms. Heterofermentative means it's produced more than one end product such as lactic acid, acetic acid, carbon dioxide, ethanol. The acid environmental between 0.6 to 0.8 percentage of lactic acid inhibit the non-lactic acid microbes and favor the growth of lactic acid bacteria. The limiting the undesirable microorganisms, enzyme that might softening the cabbage strut and creating the anaerobic atmosphere and prevent the oxidation of ascorbic acid, darkening of natural color of cut cabbage, and stimulate the growth of lactic acid bacteria. If the acid reaches 1%, it inhibits the levonostroc mesenteroid itself. It occurs in the days between 4 to 6 days. Acid 1.6%, pH below 4 allow the growth of Lactobacillus plantarum, and the final product of sauerkraut acidity is 1.7%, pH is between 3.4 to 3.6. The major end product obtained by the sauerkraut fermentations are lactic acid, acetic acid, carbon dioxide, and ethanol, and the minor end products are diacetyl, acetaldehyde, sulfur component, and ethyl butyrate. Defects and spoilage of sauerkraut fermentations are discoloration due to the autochemical oxidation, loss of acidity, off flavor and odor formation due to the growth of molds and yeast, and rancidity formation, slimy formation on the surface of sauerkraut, softening kraut or pink color kraut formed due to the aerobic growth of molds and yeast. The health benefits of sauerkraut. Sauerkraut is a high source of vitamin C and vitamin K. Sauerkraut is very good source of dietary fiber, folate, iron, potassium, copper, and magnesium. Fiber and probiotics of sauerkraut improve the digestive system and promote the growth of healthy bowel microbes, protect against the many diseases of digestive tract. A specific cabbage variety or cultivar should be used for the fermentation of sauerkraut, as well as the harvesting time is also important to obtain the good quality of sauerkraut products. In conclusion, start fermenting the sauerkraut or vegetables, start to consume fermented products, and improve the gut microbial population and improve the immune system. It will lead to prevent the infection and provide a healthy life. Eat fermented food, stay healthy. Thank you for watching. Thank you.